Okay, today we're going to start talking about membranes, the membranes that make up these different boundary surfaces that define the living cell and support the cell in several different important ways. So membranes are obviously physical boundaries that help separate two different spaces, for example, the inside of the cell and the outside of the cell. But spaces within the cell are also defined by membranes that make up the different organelles, things like the nucleus or vesicles or mitochondria, or even the ER, the endoplasmic reticulum. So today we'll start off just by appreciating some of the basics of how these lipid membranes are made in our cells, their structure, and also talk a little bit about how things can be part of the membranes as part of these different membrane functions. So at this point in the semester, hopefully, we've built up an appreciation of the fact that the living cell is made up of these different types of common components, small molecules that are individual building blocks of larger molecules, right? And in many cases, those are things like polymers, where you have individual, well-defined, repeated small units that can be chained together in a certain way to make a big, large macromolecule. In other cases, right, there isn't really a well-defined single small unit. It's more like a theme of, say, let's say, just having a whole bunch of carbons chained together. So that's how we've been learning about the four main sort of macromolecule groups built up of these components. So to start off with, let's start from the right side here. We've learned about how nucleotides that consist of a nitrogenous base, such as what's pictured here, put together with a sugar ring and a phosphate group, right? These nucleobases can contribute to the creation of nucleotides that then get chained together as long polymers to make DNA and RNA. We've also learned that the DNA contains, due to its sequence of these nucleotides, information, right? Information that can be transcribed or read from that DNA in a region of, let's say, one of our chromosomes that represents a gene. Right? And then that RNA that gets made from that gets edited, made to a finished kind of message, the messenger RNA, which then can get translated. And in the process of translation, you're making proteins, right? So every 3 bases of the RNA gets read as a triplet codon that codes for, way on the left over here, right, the sequence of amino acids that need to be put together by the ribosome that is translating the mRNA. Right? And so these amino acids come together to make proteins. So the other critical components of our cells are things like sugars, for example, right? Sugars that are used for energy, which we will learn about, for example, glucose. We already talked about how simple sugars like glucose, galactose can come together to form disaccharides, which is lactose, right? Or even long chains of monosaccharides like glucose coming together to form things like glycogen or starch. Or in plants, the production of cellulose, which is still a chain of glucose molecules, but just using different glycosidic bonds. Importantly, this is sort of like where we begin to see how these different groups sort of overlap, because after all, those simple ring sugars, the 5-ring sugars, like ribose and deoxyribose, are a critical part of being put together with something like the nitrogenous bases to make the nucleotides. Likewise, sugars are an important part of molecules like glycerol. Glycerol, which then gets put together with fatty acids. Right? So remember, fatty acids are an example where there isn't really a one well-defined single small building block. I mean, they're showing a structure here, but you can see this structure is highly variable, right? A chain of carbons, any number of carbons in here. So therefore, it's not like the same well-defined little unit as something like an amino acid or a ribose ring sugar or a glucose ring sugar. Okay. So fatty acids then can be put together with glycerol to make things like lipids. Okay. So the important thing to keep in mind here, as we sort of think about these themes of these molecules, these different groups, all being equally important in our cells, right? But they're not equally sort of coded for by our DNA, because after all, our DNA really just codes for RNAs, right? That may or may not just function as RNAs or be translated into proteins. So it's the proteins then that then handle things like fatty acids or sugars to be able to take those types of building blocks to make things like complex carbohydrates or things like lipids, right? So that's one way of thinking about it, right? That ultimately what's coded for in our genome are the information to make the proteins. Things like the carbohydrates, the fats, things like that, they are not actually coded for by our genes, right? Our genes make the machines in the form of proteins that can then handle or make use and manipulate things like sugars or fatty acids, right? Thereby making those things as a function of the proteins doing their job. Right? So you don't have a gene somewhere that codes for glucose, or you don't have a gene somewhere that codes for a particular fatty acid, right? It's all up to the proteins to handle and make those things, right? Either from scratch inside of your body using the materials that you already have, or by stuff that we have to eat and take in from the outside, right? So certain types of lipids and fatty acids we will learn, right? We cannot produce our proteins. We don't have the right machines to make these things, so we have to get them from our diet. All right, so let's focus on things like the fatty acids and lipids today, because we're talking about membranes. So any discussion of membranes has to start with us kind of going back to square one and thinking about the importance of water and intermolecular water interactions, right? And I know some of you are like, back to water again, come on, we beat this whole topic to death. But water and this concept of how water influences how the molecules in living things work is absolutely fundamental and essential for you to be able to appreciate why biological molecules are shaped the way they are and how they're able to interact in the way that they do, that life depends upon. So remember, all the way back from the beginning of the semester, we said, yeah, water, H2O, right? One red oxygen with two white hydrogens in this model here. And it forms this little polar molecule. Polar because the oxygen tends to be more greedy with the electrons. It has more pull. You know, after all, it's sharing these electrons with the hydrogens, right? But the poor hydrogens are basically just a proton with a little a proton with an electron that it's going to share with the oxygen here, but the oxygen is pulling the electrons more towards itself, and that's what makes the water molecule polar, right? So you get sort of like a little negative charge on the red side and more of a positive charge on the white side, because you have the two sort of exposed protons on this side, if you want to think about it, right? So we said that substances that you try to maybe dissolve in the water, like whatever this compound is that has this green atom and this hydrogen attached to it, it may be hydrophilic, meaning this molecule itself has kind of an imbalanced positive and negative characteristic to it. And as a result, it plays very happily with the water molecules, right? The sort of negatively charged green part of it tends to attract the positively charged hydrogens of the water molecule. And likewise, the hydrogen part of this compound, whatever it is, happily attracts the red oxygen parts. As a result, we say it's hydrophilic because the water loves it, right? The water surrounds it, and whatever this compound is easily dissolves because these individual molecules, float off into the water. Right? Hydrophobic substances, on the other hand, like this here, right? So let's say maybe this black atom is a carbon and it has 4 hydrogens around it. So this is like a CH4, like a methane. The distribution of the charges is very even, and there is no sort of net positive negative to it at all. So as a result, the water molecules have no interest in being attracted to it. So all you have are, if you dump this thing into water, right, all the water molecules are just bumping into it, right? They don't want to stick, they don't want to interact, they're just shoving all these methane molecules around. So naturally, what would happen then, as these individual methane molecules are getting shoved around, they will tend to group together. Not because the methane molecules are somehow attracted to each other, right? That's the false way of thinking about it. It's not that the methane molecules are attracted to each other, it's just they have nowhere else to go because the water molecules keep bumping into them. And so the most convenient, natural way that the water molecules will shove them is they will shove them together. Right? And this has to do again with the property of water and all of its hydrogen bonding sort of properties, right, with each other. Right? This is the same principle, ever since you were a little kid and you were eating at breakfast a bowl of Cheerios, right, in a bowl of milk, and you look down and you see that the little Cheerios that are floating around magically seem to like come together, right, to form these little rafts of Cheerios, right? And it's fun. You mix it around in the milk, and then eventually they all stick together, and you think, oh, right, the Cheerios are attracted to each other, right? Well, now you know it's not that the Cheerios are attracted to each other, right? Because why would there be some sort of magical magnetic pull between Cheerios? No, it's because they're all getting shoved around by the water molecules on the surface of the milk, right? That's your sort of surface tension. And those molecules just shove the Cheerios around because they don't want to interact with them. And the most convenient place all the Cheerios end up getting shoved is together. Right? So why is that important when we talk about lipids in the context of lipids making up things like membranes? Because even though we think of membranes as being physical solid barriers, in reality, right, they're not very solid at all. What do I mean by this? The lipid bilayer that forms the surface of, let's say, a cell membrane, I think most of you realize, is actually a bilayer, right? It's two layers of lipids that are kind of brought together. So in this illustration here, all of these blue and red balls up here with these sort of greenish tails sticking down, these are individual phospholipid molecules. Right? And we'll take a closer look at their composition. So you have a layer of phospholipid molecules on the top here, and then you have a layer on the bottom, and all of these fatty acid tails point to each other towards the middle. Now within this, you have a rich diversity of other types of molecules kind of suspended in this lipid bilayer. You have these large orange things, which are proteins, right? So many types of proteins function and are meant to be sort of anchored to this lipid bilayer. Some of these proteins, remember, these proteins are just chains of amino acids that have now folded up into a particular shape, right? Some of them form shapes that form like channels, right? So there's a cross section of a complex protein that has a channel that allows the space from the outside of the cell to be continuous with the space that's on the inside, right? Passing through the lipid bilayer. Other proteins like this one here might serve as some sort of receptor protein. So part of it is filled with hydrophilic amino acids. So therefore, this part of the protein naturally is happy sticking out into the water up here. Remember, all of this white space in the slide here is not blank, right? It is filled with water molecules. We just don't bother showing it because then you wouldn't be able to see anything else, right? So the water molecules play happily with hydrophilic side chains of amino acids that make up whatever chain of amino acids is represented here in orange, whereas you have other amino acids that are a part of this peptide sequence that are hydrophobic, right? So as a result, the water molecules, if this was to stick out, right, the water molecules would just shove it back in to this hydrophobic space where it can happily kind of hang out, right? And many of these proteins are modified with things like these sugar groups. So these little green things are maybe like glucose or galactose, And so you'd have these different structures, chains of oligosaccharides, right? So oligosaccharide is like a polysaccharide, except for it's not super long. Some of these are maybe only like 6 or a dozen sugars, so you can call them an oligosaccharide, right? Not a mono. It's not big enough to give it credit for being a real big polysaccharide. It's sort of a short stretch oligosaccharide. And these then help alter the shape of different proteins so that these proteins might serve different functions. But I want you to think about this. The membrane of the cell, right, is riddled, speckled with all of these types of proteins and other types of molecules. So it's like a mosaic, right? A mosaic, like a mosaic of tiles shown up here in the corner here, except rather than being a rigid, like tile piece of art, like you see here, this thing is fluid, right? Because it's just these lipid molecules, phospholipids. And this is the real thing that I think, when I first realized this many years ago, kind of blows my mind, right? What keeps this together? You might say, well, you have these 2 layers of phospholipids, so they're kind of attached to each other, right? And that keeps us together. No, they're not, right? They're only held together there, right, in a similar way, like the Cheerios are held together in your breakfast cereal. Right? It's all because of the surrounding water. If there was no water here, right, this whole thing would just fall apart because there would be no reason, no forces keeping these things together. Right? All right, so let's talk about these lipids that make up the membranes. So lipids are made of fatty acids with other groups attached to them. So we've talked about lipids before, early when we were just sort of introducing them as being one of the important macromolecules. Let's take a closer look. Right, some of these things we already discussed, but I want to make you think about it in a different, slightly different way now, a little bit more advanced. Right? So fatty acids, this is the structure of a fatty acid. So you'd have a long chain of hydrocarbons, right? So here I say CH2s, a chain of CH2s, but the reality is the reason I only write CH2 is to make sure you realize it's not just carbons, right? It's carbons associated with hydrogens on them. It doesn't have to be 2 hydrogens, but I just wanted to remind you that it's not just a naked chain of carbons, right? And then on one end, you would have the last carbon would probably have 3 hydrogens on it, right? Because remember, each carbon atom can make 4 bonds. So if one of the bonds is the carbon-carbon bond that's attaching it to the end of the chain, then the other 3 potential bonds it can make, it'll happily make with hydrogen, right? So you'd have a methyl group at the end here. And then the other end, you would have a carboxylic acid, or this would be a carboxyl group, a carbon double bond to oxygen, and then bond to an OH, right? So this structure is what we call a fatty acid, and it's highly variable, right, in size. So that's why it doesn't really fit the term of this being a well-defined, like monomer or something like that, right? Like an amino acid is very well defined, a single sugar is very well defined, a nucleotide is very well defined, but a fatty acid, it's kind of like whatever. It's just a big chain of hydrocarbons with a carboxylic end on it. So here's some examples of fatty acids. These are fatty acids that are actually quite important in our diet. So stearate is listed here. Okay? And so the nomenclature of fatty acids is typically something like this. So stearate is denoted as being 18:0. Right? The first number, the 18, refers to how many carbons are involved in this big, long fatty acid chain. Right? So if you were to count this out, you count 18 carbons involved. The next number, the 0, refers to how many double bonds are present in the actual carbon-carbon chain. Right? So we're not counting the double bond between the C and the O in the carboxyl end, right? Because all carboxyl groups would have that. We're talking about in the main chain here, how many double bonds are there? In this case, 0. Right? So as a result, stearate is very linear. Because if you look at this here, all the carbons are black and the hydrogens are blue, right? So this fatty acid is saturated. Each carbon is perfectly happy. It's satiated in a way, right? Satisfied with having all of its bonds filled up with hydrogens. Right? Let's look at oleate. So oleate would be denoted as 18-1. So again, it's like the same 18 carbons that you have in the stearate, but now you have one double bond in it over here. All right, so that means the structure has 2 fewer hydrogens in it. Right, so the hydrogens had come off of these carbons, and as a result, these 2 carbons can form a double bond with each other to give each of them their happy 4 bonds. Right? And that, of course, makes a kink in the structure of the molecule. Right? So as a result, at room temperature, compounds like this one up here would tend to more happily settle together, right? Because the amount of energy at room temperature, right, is low enough that they'll settle together, and you might have a solid, whereas a compound like this, right, at that same temperature, the same amount of energy, because they're kinked and bent, they won't stack up. And so this particular fatty acid might remain liquid at room temperature. And then here's a third example, again, made up of 18 carbons, but now you have 2 double bonds. And so this particular fatty acid we call linoleate, right? And so you might say, well, this is just like, getting too detailed, because who cares? They're all 18 carbons and they're pretty much the same, except for they have different slight bends in them, right? Why bother going to the trouble of giving these things different names, right? We bother doing it because these different shapes that they have mean that only specific proteins in our cells are the right shape to interact with them, right? And therefore, these 3 molecules, even though they look very similar compositionally, right, and all 3 of them are 18 carbons, they have very different uses inside of our cells, because they can bind to different proteins that are evolved as machines to specifically recognize either a straight chain or one that has a slight kink or one that has more of a zigzaggy kink, right? So let's think about this. Here's an example of a type of fatty acid that maybe you've heard of more. You might have heard of linoleate and stearate or oleanic acid, something like that. But omega-3 fatty acids you hear about a lot, right? Those of you who have any sort of pay attention to your diet, you read about things like omega-3s are good, right? You want something that's rich in omega-3. So what's an omega-3? So these omega-3s are very important for our cellular metabolism and also for the production of our membranes. They serve as the raw material for getting a lot of fatty acid components to make things like membranes. This is the structure of an omega-3. So you have the carboxylic end over here, the carboxyl group, right? Then you have a chain of carbons, so hydrocarbon chain. And again, this could be variable, right? So it does not necessarily mean that this is the exact has to fit this particular picture here, right? But you have a variable chain of hydrocarbons, right, that ends with the methyl carbon at the end, the CH3. So we call this the alpha end, the carboxyl end, and the omega end, right, the CH3 end. So in omega-3 fatty acids, the 3rd and 4th carbons from the omega end is where you would find a double bond. Right? There may be double bonds elsewhere in the chain, but the important one here that defines this as being an important omega-3 is this double bond at the 3rd position from the omega end. And that's why it's called omega-3. Why? Because again, we evolved to have certain proteins that are just the right shape to recognize a fatty acid that ends 3 carbons from the end with a double bond that puts a kink there, right? That's why it's important. So these come in from different sources in our diet. Importantly, our body cannot make these omega-3 fatty acids from scratch, right? So we evolved to expect to get these omega-3 fatty acids from our diet. And then we have proteins that recognize that these are omega-3 fatty acids because of that double bond at the end. And those proteins can then go to work and utilize these to make other components in our cells. So here's 3 examples. ALA is what we call an alpha linolenic acid, and we get it from plants. It's an omega-3. So things like nuts, things like that. EPA and DHA come from oils that come from things like seafood, right? So you often hear about how fresh fish is important as a source of omega-3 fatty acids, right? So that's what we're talking about. Okay? So this is an interesting point. Here you have those two fish-derived omega-3s. So EPA is eicosapentaenoic acid, DHA is docosahexaenoic acid. You don't have to remember the names of those, right? If you just remember it as EPA, DHA, I'd be happy, right? Because I can't even pronounce them. But here are the structures. So you can see here's the alpha end with the carboxyl group, and you go all the way to the end. This would be the omega end, and the 1, 2, 3, and 4 carbon from the end has a double bond, right? So that's what defines these as omega-3s. So a fascinating kind of thing to think about here is because these are filled with double bonds, these are particular bonds, double bonds, are targets for oxidation. In other words, if you have oxygen present, right, that can actually get its way in between the double bonds and form the double bonds to break. So you end up with the oxygen just being added to one of the carbons on one side, and then the other carbon that was part of this double bond, you know, whatever is hanging on to it gets separated. And so because of the third double bond at the end, these omega-3s oftentimes have bonds broken in them that release, right, a small part of it. And if this part of it over here, the alpha end, gets released due to oxidation, in other words, oxygen coming in and breaking this double bond, you have the release of this molecule, right? And this is butyric acid. So butyric acid, the name butyric acid actually comes from the word butter. Right? Butter itself, I guess a more accurate way of saying it is, but the word butter itself comes from the Greek word for butter, right? And so butyric acid is derived from that. In the early 1800s, there was a French researcher who was working with different types of lipids coming from food products, and he isolated butyric acid from butter, and he realized that the butyric acid had that buttery smell to it, right? So the smell of butter that we love so much is our nose picking up on free butyric acid molecules that are floating around. Now, if you have a nice fresh stick of butter, right, it's a pleasant smell. But what happens when the butter goes bad? It smells really bad, right? What is that? It just means that so much of the lipids are breaking down in it that you have the release of so much butyric acid that it kind of gets sensed by our nose as no longer being pleasant, right? And it triggers bad smell kind of perception in our mind, right? So that's the same thing with these omega-3s that come from fish, right? If you have a lot of oxidation of these, it releases a lot of butyric acid, and that gives us that kind of, ooh, stinky fish smell, right? Or the smell that we associate with rotting food. Why is it unpleasant? Because after all, I mean, whatever, look at the butyric acid, right? There's nothing special about it. It just came from this lipid. So why is it something that in small amounts we like the smell of, but we evolved to not like the smell of a lot of it? Because, right, smelling a little bit of it evolutionarily means, you're coming across something that is rich in fat, right? So that makes you want to eat it, like buttered popcorn, right? But if you encounter something that has so much butyric acid coming off of it, that probably means that thing has gone rotten, right? Because bacterial activity on there, right? The bacteria, if this is a piece of meat in the sun, right? That accelerates the oxidation of these lipids, releasing the butyric acid, right? So that's really interesting to think about. A lot of times, things that we smell with our nose are small, little organic compounds like this, right? And depending upon evolutionarily, if it's a good thing or a bad thing, that's how we have evolved to interpret it as a pleasant smell or an unpleasant smell, which might be a warning, because you can imagine, right? Mammals that couldn't smell the stinky rotten fish and ate the stinky rotten fish, they tended to die more from food poisoning, so they didn't pass their genes on, right? But organisms that could smell the stink and not want to eat that particular piece of fish, they had a better chance of passing their genes on. All right, so I said that things like omega-3s that we get from our diet are important raw materials for the proteins in our cells to then break apart and actually put together again to make things that we need, right? Like phospholipids. So phospholipids are the main component of our cell membranes. So this is the structure of a phospholipid. Well, let's start with the cartoon over here, the least kind of threatening part of the slide, right? So a phospholipid has two fatty acid tails hanging off of it and a polar head group, right? This would be what you see on the outside of the membrane, right? And then the fatty acid tails stick on the inside. So there's three parts that make it up. So you'd have the two fatty acid chains here. Right? Then you would have a glycerol group. So a glycerol group is 3 carbons. It's an alcohol. And you'd have the addition of a phosphate group. So when you put those 3 pieces together, this is what you get. Right? So the glycerol in many ways acts as kind of like a staple that holds together the 2 fatty acid chains, and then the 3rd carbon on the glycerol, instead of holding another chain, I mean, it could, but if this was a glycerol that had 3 fatty acids coming off of it, it would not be a phospholipid in our membranes anymore, right? That would be a triglyceride, right? That would be the type of fat that your body makes to store extra energy, right? But in this case, if the 3rd carbon in the glycerol has a phosphate group attached to it, right, this makes a phospholipid. And importantly, that phosphate group is quite capable of having some other random R group, attached to it, whatever that might be. In this particular case, the structure that you see here with these 2 carbons and then this nitrogen with the 3 methyl groups around it, this R group is choline, right? Choline that together makes this a phosphocholine, right? That attach together. This is the most common type of phospholipid that you would find making up the walls of your cells. The choline group itself, right, this group over here, is also used for making different other things, like neurotransmitter molecules, like acetylcholine, right? Okay. So as a result, this group, the R group, the phosphate group, because of its charges, right, is highly hydrophilic. Water molecules would love to play with this end of the molecule and form little hydrogen bonds and interact with it, whereas the fatty acid tail end, water molecules, they have no interest in it, right? Because there's no charges to interact with. So the water molecules are just bumping against these, shoving the tails kind of out of the way. So as a result, right, if you had a bunch of these molecules together, right, sort of floating around, let's say like the Cheerios in your milk, right? The Cheerios got pushed together by the water because there was no place else to go. These molecules, these phospholipids, would also be pushed together. But when they get pushed together, the blue part of this cartoon, the polar heads, would face out, right? Because they're still happy playing with the water molecules, but the fatty acid tails would get pushed in to be together. Right? So you'd form like a little bubble that just has the blue polar heads on the outside and the fatty acid tails in the middle, right? But if you have a lot of these molecules, it doesn't just have to be a little bubble. It could actually form a complex shape like this, right? Where you end up with 2 layers of phospholipids. Each side off to the left and to the right is playing happily with water molecules, but the fatty acid tails are just shoved away by the water. They have no place else to go. So the best place to go is just towards the inside of the membrane, right? And again, think about that, right? That this is not held together by bonds between these different phosphate lipid molecules, right? They're just positioned there because that's just the most energetically place they get shoved, being surrounded by the water. Right? So look at this illustration or this animation here. That's what this really depicts, right? These are all these phospholipid molecules that make up our bilayer, and the fact that they're kind of jiggling around, right? It's kind of weird. I'm like staring at this like, okay, that's kind of weird. But that reminds you, they're not actually bound to each other, right? They are just there because they're surrounded in the white space by all these water molecules shoving them there and flipping them in that orientation, where the hydrophilic ends, the head ends of these molecules, stick out because the water molecules play with them. the hydrophobic ends, the fatty acid tails, right, get shoved in there. Also imagine, right, that you can mix in here big protein structures, big protein molecules that would also be in here, right? And what keeps this stuff together? It's the water. So if you have a protein molecule in here, if the protein molecule started floating up or started floating down, right, the water molecules would just shove the hydrophobic parts of that protein back into the membrane. So in a sense, I think that's kind of freaky to think about, right? That you are in fact not very solid. You are held together by water molecules holding you together. If it wasn't for the water surrounding your cells, none of this would stay together, right? Your cell would not be a solid thing. All the molecules would just disperse. So your cell is held together by water. Therefore, all your tissues are held together by water. Therefore, you sitting there is held together by water. That's how important water is for life on Earth, right? And that gets you thinking, because not all planets in the solar system, you know, or in the universe even, would have liquid water like we do. All right, let's elaborate a little bit more on the structure of different variations of lipids that can make up membranes. So here we have a group of lipids known as glycolipids. So the moment you see glyco, you know that has to do with sugar. Right? The simplest type of glycolipids would be this one on the left here, where you have some kind of a lipid, right, fatty acid tail or whatever hanging off, and then at the end of that fatty acid tail, you have a sugar at the end. So in this case, here's a sugar ring, right? So this would be a simple glycolipid, all right? Nothing really earth-shattering about that, right? But it can get a little fancier, because what if you had two fatty acid tails that are stapled together by a glycerol, just like you were making a phospholipid, like we were looking at on the previous slide, but instead of a phosphate group on the glycerol, oh, you have a sugar instead, right? So instead of a phosphate group, you have a sugar group. Well, there you go, right? That's not too hard to understand either, right? So this is what we would call a glyceroglycolipid, right? So very similar to a phospholipid, except for instead of the phosphate group, you have a sugar group attached to it. Right? Then you can get a little bit weirder, because in this case, what you see is you have a fatty acid tail, a big long chain that kind of goes off somewhere. And you have a sugar group, the sugar group at the end here, but you have this purple structure circled here, right? This structure that is a chain of carbons with a nitrogen group on it or an amine group on it, and then a chain of 12 hydrocarbons ending with a methyl group that has a C and 3 H's. This is what we call a sphingosine, right? And so this type of a structure would be called a sphingoglycolipid, right? And you're like, that sounds kind of random, right? Because obviously there's many variations of other ways you could put together a fatty acid, a sugar, and something in between. Why is this sphingoglycolipid worth talking about? Right? Because these are found on the surfaces of our red blood cells. So our ABO blood type, right, is determined by what type of sphingoglycolipid you have on the surface of your blood cells. Right? So on the top here, again, we have that sphingoglycolipid. We have a fatty acid tail and this weird sphingosine group, right, in pink here. And then you have a sugar, right? So that's your basic sphingoglycolipid. So on the surface of our red blood cells, we have different variations of this. You can see here, you have a sphingoglycolipid that has an oligosaccharide attached to it. This is composed of simple sugars, glucose, galactose. This is what we call an N-acetylated galactose. So it's a galactose that has an acetyl group with a nitrogen attached to it. Then you have a galactose and you have a fucose, right? A different 3-ring sugar there, right? In our bodies, our red blood cells make this particular sphingoglycolipid on their surface. So their whole surface is studded with these. These are floating around in the membrane. So the fatty acid part of it would mean it stays anchored in the membrane, because that fatty acid tail, if it ever floated out of the membrane, all the water molecules would just encourage it to go back in the membrane. But then you would have this sugar, the short sugar group sticking out, right? In the human genome, we have a gene, which we now just name or refer to as the ABO gene, that codes for, right? It codes for a protein that is a sugar transferase. In other words, this protein can bind a sugar and add that sugar to something else. Right? The functional form of the protein, right, can grab onto things such as N-acetyl galactose molecule sugars. Right? And so if you have a version of this gene that makes the protein that is the right shape to take an N-acetyl galactose group, it will also, of course, that protein will also recognize the shape of this galactose and fructose sugar sticking out. It'll bring them together, right? And it'll add that N-acetyl galactose group to the end of this oligosaccharide, right? And if that happens, the resulting shape is what we call the type A antigen. That same gene that codes for that same protein, some people in the population have a slight difference in the genetic sequence, resulting in, I think, 4 amino acids in the finished protein being slightly different. As a result, that version of the enzyme can only bind to just plain naked galactose, not the N-acetyl galactose. But it'll happily then add that version of the sugar to the end. And if that's what you get, this shape will be slightly different than this shape up here. We call this the type B antigen. And interestingly, some people have a frame shift mutation in the coding sequence of the gene, so that because of a frame shift, they end up still successfully translating a protein from the mRNA, but the mRNA has coded for something completely different because of the frame shift. That version of the protein cannot bind or transfer any sugars at all. So as a result, people with that version of the gene don't get anything added to their red blood cell antigen. So that's type O, right? So about 44% of the population, roughly, right, are type O. About 42% of the population are type A, right? And something like, what is it, like 10% of the population are type B. Right? Now remember, you have 2 copies of the genes that do this, one that came from mom and came from dad, right? So if both of them are type O, right, from mom and dad, right, then none of your red blood cells, you won't have any of the protein that can successfully add sugar groups to these, so you'll be type O. Right? But if one of your parents gave you a type A or a type B, right, then that means your body is making the protein that can make this modification, right? So you would be type A or type B. If you got a type A allele and a type B allele from mom and one from dad, then you would be type AB, right? Because your red blood cells would have a combination of some of the antigens looking like this and some looking like that, okay? So again, right, the interesting thing about this is to this date, we don't know really the physiological significance of why we evolved to have different blood types like this. I mean, look, there's got to be a reason, right? Because if there wasn't a reason or some advantage or whatever, then why would this be kept as part of the way our human genome works? The interesting thing is, I mentioned to you, the type O people, right, their version of the gene, because of the frame shift, the protein that gets made from it is very different than the type A, type B transferring protein, right? But we don't know what that protein does. But again, it's fascinating to think about. So as you read about different types of public health studies that maybe have some data that say people who, are infected with certain maybe viruses like COVID, maybe over time, you'll read some reports that say, oh, type people, react a certain way, or type A and B. It's interesting to think, right, that maybe there's an evolutionary reason behind that in terms of how we evolved to be able to cope with things like viral infections. All right. Another group of membrane lipids are the sterols. So sterols are long chains of hydrocarbons as well, but in this case, right, they're wrapped up as rings. So just because something's in a ring doesn't mean it's no longer a hydrocarbon, right? Because I know we tend to think of hydrocarbons as long chains, but hey, right, things can wrap up into a ring structure and it's still a chain that's composed of carbons. So this 4-ringed structure is known as a sterol. Right? It's the basis of things like making the steroid hormones in your body. So this structure here is cholesterol, the 4 rings of a sterol, and then this chain of hydrocarbons coming off. Right? Cholesterol is a very important component of our cell membranes. You know, cholesterol gets kind of a bad rep, right? When you think about cholesterol, it's always like, ooh, stay away from cholesterol. That'll give you heart disease, or fatty foods have a lot of cholesterol. But cholesterol is actually a vital, essential part of your cell's composition. You can see the cholesterol molecule here interacts through hydrogen bonds with the phospholipids. So as a result, throughout your phospholipid bilayer, you have these cholesterol molecules in there. And that actually helps. It helps maintain the proper kind of spacing and structure of your cell membrane. As I mentioned to you, right, cholesterol is also the starting point for your body making things like steroids. Steroids that control inflammation, like glucocorticoids, for example. Cortisones, things like that, are steroids, right? And when we think about steroids, I think we typically think of the sex hormones like estrogen or testosterone, right? Those are all derived from cholesterol. Okay, so this kind of summarizes the 3 main groups that we talked about, right? Phospholipids, glycolipids, and sterols are like the 3 important lipid classes that make up our cells, right? So this cartoon helps you kind of keep that in mind. So the proteins then. We will talk more about proteins in membranes with the next lecture, but I wanted to introduce this concept here, right? In purple are these proteins. Remember, these proteins are just long peptide chains. Many types of proteins associate with the membrane because, as part of their amino acid sequence, they would have more hydrophobic amino acid side chains in these parts of the protein, and therefore the water that's surrounding all of this would tend to shove these into the membrane and keep this anchored there. Right? Some proteins are drawn to the membrane and hang out by the membrane, not because they interact with the membrane itself, but because they interact with other proteins that are there. Right? And lastly, some proteins have post-translational modifications done to them, like the addition of fatty acid chains to them. And that provides a little anchor to keep them stuck to the membrane. Right. And as I mentioned before, some of these protein structures might serve as channels, and that's something that we'll explore next time. Right. Others can be heavily modified with sugar groups and act as some sort of signaling shape, right, that allows cells to communicate with other cells. Okay, so I just wanted to end today with something a little fun because we're almost out of time. You know, I try always to kind of draw in some things from daily life that you guys can relate to kind of help you see that this stuff we talk about, you don't learn it just because I need to learn it to pass Bio 275. No, a lot of this can be really fun if you see how it applies to stuff around you. In different types of plants, especially corn, that produce a lot of starch. Remember, starch is just a big chain of glucose molecules. Oftentimes, the starch from things like corn can be enzymatically modified to turn them into little rings, right? To make things like what we call a cyclodextrin, because individual glucose is also called dextrose. Or you might not have known that, but dextrose is the same thing as glucose, right? So here you have a ring of glucose, right? So this is a cyclodextrin. Now you draw it this way in 2 dimensions on a piece of paper or the screen, but of course, right, this whole class is about realizing that things are actually in 3 dimension. So the natural way that this ring would want to kind of get comfortable is if the hydrophobic surfaces of each of the rings kind of gets shoved towards the inside, and the hydrophilic parts, like different groups, like the hydroxyl groups that are sticking off, can play nicely sticking out. Right? So you'd end up getting this sort of like barrel shape here. Right? And what's interesting about this barrel shape, it is like a little barrel, right? The outside of it can be surrounded by water happily because the water will interact with the hydroxyl group sticking out, but the inside of the barrel is very hydrophobic, right? Almost like the hydrophobic interior of our cell membrane. So cyclodextrins have many useful applications. Because they're very small, they can actually be used to help contain or trap other small hydrophobic molecules. and protect those molecules. So as a result, if you have certain pharmaceutical drugs that are small hydrophobic molecules that might otherwise be sort of like destroyed or metabolized by your body, right? They can be packaged up in these types of cyclodextrins so that they are protected from hydrolysis by water molecules. Many types of pigments like colors and dyes, things like that, right, are actually formulated in combination with cyclodextrins to preserve the color. Why? Because lots of small ringed molecules like this one here, this is like a benzene ring, right, with a carboxyl group on it. It's the ring structure that gives different compounds colors, right? Because wavelengths of light that bounce around in the ring bounce back in certain wavelengths, right? So to protect these rings from being destroyed by water doing hydrolysis, right, you can package them up, and therefore dyes stay brighter and more colorful. Also, food products, right? Because as we mentioned, many of those aromatic good smells, things like butter, are these types of hydrophobic molecules that would otherwise get broken down and destroyed in water, right? So many types of food preservatives are cyclodextrins to kind of trap these pleasant aroma scent molecules, right? Oh, and I should mention to you, these ringed molecules, right, tend to be the types of small molecules that we can smell very easily. And that's why they're called in organic chemistry, what? Aromatic compounds, right? Aromatic compounds are small ringed compounds, and they're called aromatic because long time ago, before we knew any better, we just said they smell nice, they smell sweet, they smell pleasant. Sometimes they don't smell pleasant, right? So this is the other application. These cyclodextrins can be used to eliminate odors, right? Like, oh, stinky stuff that comes from fish. Wouldn't it be nice if you had stinky, aromatic rings of compounds in the air, if you could somehow trap them, right, in these cyclodextrins, so that then when you breathe it in your nose, right, those aromatic compounds don't interact with your olfactory smell receptors, right? That sounds like it could actually be a very good invention, right? Well, I hate to break it to you, right, but they've already done that, right? Because that's what Febreze is. So you all know Febreze, right? You spray it around when you have a stinky couch or stinky gym clothes or whatever, right? But maybe you always thought, Febreze, that's just like some perfume or something that covers things up, right? No, it turns out Febreze is made of cyclodextrins. If you look at the ingredient on the Febreze bottle, the very first ingredient is water, right? Because you need water so you have something to dissolve it in. But the second ingredient actually says cyclodextrin, right? And it says odor eliminator made from cornstarch. Right? So when you spray Febreze and it comes out in little droplets of water with the cyclodextrins in it, then these aromatic stinky molecules that are floating through the air, right, get stuck into this water droplet, and then they have a chance of the water molecules to shove the stinky molecule into the little barrel of the cyclodextrin. And now when you breathe it in, you no longer smell the stinky molecule. Right? And then this is cool because this is actually a three-dimensional image of the molecular structure of Febreze. Right? Here's the barrel and then rotated to the right. And here is an aromatic compound that is being shoved in there by the water molecules. This water droplet that has the cyclodextrin in it that now also has the stinky molecules absorbed by it. Right? So really cool stuff you can do if you understand basic molecular cell biology principles. Right? With many applications for different things like industry or even stuff like this, right, that you might not think has anything to do with studying cells. All right, let's go ahead and stop there today.